Good afternoon, everyone. I recognize a lot of names from people I've worked with in the past or at present. So I was asked to talk about a couple things today. One was to talk about building an interdisciplinary research team, and the other was to talk about integrating mobile technologies. Since I happen to be a pain researcher, I actually will tell you a little bit about chronic pain in this talk. So there'll be sort of three parts to the talk. A little bit about team building, a little bit about gadgets, and a little bit about pain. My disclosure slide. So I'm glad Danielle didn't introduce me the way people usually read an introduction, because it sounds like if you would read a short bio of mine, it would seem as though I was born a researcher or became a researcher in utero, because I have been fortunate enough to have a successful research career. But I started as a pure clinician. I went to undergrad in medical school at the University of Michigan. Was intentional about deciding to do my clinical training at Georgetown University because I wanted to really learn clinical medicine quite well, and Georgetown was a terrific place to learn clinical medicine. And so I was trained in internal medicine and rheumatology, and never really thinking that I was going to go into research. And then when I was in my final year of rheumatology training, I was moonlighting in an intensive care unit and reading the Washington Post, and saw a little article in the Washington Post saying that the state of Minnesota had removed the nutritional supplement L-tryptophan from the market in the state of Minnesota because, as part of some state health department surveillance they were doing, they had seen that a whole bunch of people that were taking tryptophan had developed a high eosinophil count. For those of you who don't practice clinical medicine, eosinophil is a certain type of white blood cell that stains red, but it's pretty unusual. Eosinophils are very high in allergic conditions. But what struck me was that we had seen a bunch of patients, not a bunch, three or four patients at Georgetown over the past year or two that had a really high eosinophil count, and we really didn't know what was going on with them. And we just sort of gave them supportive care. So after I read this article, I remembered the names of these individuals. I called the four people that I could remember seeing over the past couple of years, and even though nothing was in their medical record about them taking tryptophan, they all had, in fact, been taking tryptophan. And so then I asked the rheumatologists who were practicing in the Washington, D.C. area if they had seen anyone with high eosinophil counts. They gave me three additional names. I called those three people, and all three of those people also had been taking tryptophan. And so after doing this, I was able, as a second-year rheumatology fellow, to get a first author case series in JAMA describing a new disease that eventually ended up being called eosinophilia myalgia syndrome. But this really hooked me on doing research. I hadn't actually uh, expected to do research, and there were no formal training programs for clinical researchers at the time that this was all occurring in the late 80s, early 90s. Um, Nonetheless, I became one of the world's experts in a new disease that was called eosinophilia myalgia syndrome, and the company that inadvertently caused this problem provided a lot of funding to do research on this condition, but I didn't know how to do research. So the only way I could do research was to identify people either at Georgetown or up the road in Bethesda at the NIH that had established research expertise and that could work with me to research this new disease, eosinophilia myalgia syndrome. And I got a lot of grants and did a lot of studies, but I joke that I was an early adopter of team research and team science because I didn't know how to do research. The only way I could do it was to create teams. And so first, working in eosinophilic myalgia syndrome, we did animal models, we looked at mechanisms of action, we did clinical trials of new therapies. But again, because I was a clinician that had sort of identified this new disease, I was able to find collaborators that could help me study this condition. And then because I was very early as a junior faculty member at Georgetown, I had the most new patient slots available. Nearly all of the individuals that were being referred to me were individuals that I diagnosed with a condition that at the time we knew very little about, that is now called fibromyalgia, it used to be called fibrositis. but this is a syndrome where people have pain throughout their entire body. And at the time, we didn't really know what was causing this pain. And in fact, most people at this point in time in the late 80s, early 90s didn't even believe that these people had a real problem. Most people thought that individuals with labels like fibromyalgia were just making it up, that they really didn't have any pain. But again, I thought fibromyalgia was fascinating. I was seeing these patients. I didn't in any way believe they were making this up because these findings seemed very real. So again, I put together groups of individuals that could study the fibromyalgia and embraced the notion of the team, the team, the team, because I was seeing the same sort of fibromyalgia phenotype in people who stopped taking tryptophan and still had eosinophilic myalgia syndrome. There was a large sort of epidemic of fibromyalgia-like illness after the first Gulf War, and we received about $25 million in DoD funding to study the first Gulf War. A lot of the brain imaging studies that were just coming into being that we used to study Gulf War illness were we then used to study pain and conditions like fibromyalgia. But nonetheless, in this time while I was still at Georgetown, I was putting together teams of individuals to study different pain conditions that had an element of fibromyalgia or an element of pain that was coming more so from the central nervous system rather than from the periphery. And got the opportunity to move back to the University of Michigan in 2001. I was the first PI of our CTSA grant, the founder of MISHAR an awful long time ago. But now all I do is lead a large pain research group called the Chronic Pain and Fatigue Research Center. I'm going to talk a little bit about what you need to do team science, talk about mentoring and individuals of different disciplines, highly trained staff. This is sort of an underappreciated, important thing in team science that you need really, really good staff to do these kinds of studies. Uh, More and more, we are now involving what we used to call patients, then research participants, and now we refer to these individuals as persons with lived experience, or PWLEs. And these individuals now are often involved from when we start thinking about doing a study to as we execute and then as we finish the study and disseminate the findings. You need a good environment and protected research time. Now, there are now a lot of really good training programs, training grants. We have a lot of those grants and we love training and mentoring individuals in team science. And then you need pilot studies and funding to lead to larger and larger grants and studies. And it often is the case if you're trained as a PhD and you're not clinically trained, often you are more reliant on a clinician that comes up with a reason to do a clinical research study. Again, I'm talking now about clinical research, not NSF-funded research. More and more, the core idea that you're studying is the most important thing that's going to determine what score you get on your grant. You are going to need to identify collaborators or mentors with previous experience in doing the type of work that you're proposing to do. I talk a lot about how clinically trained individuals and PhD trained individuals are almost like vertically and horizontally positioned in team science and that they need each other badly. The PhD trained individuals often are expert in research methods and are fairly agnostic to the disease or the condition that they study. Whereas those of us who are trained clinically are confined by our clinical specialty more or less into the conditions that we study, and then we need to find people with methodologic expertise in a whole bunch of different areas. You need statisticians and or data scientists. Get those individuals involved early and often. You need nurses, medical assistants, research assistants, and then again, persons with lived experience. Many of you, if not most of you, are at the University of Michigan, and if you have never seen this video yet, you need to. If you have seen it, I'm sorry, you're going to see it again. But it's the famous "the team, the team, the team" speech from Bo Schembechler. Oops.
We're going to win it as a team. They can throw out all those great backs and great quarterbacks and great defensive players throughout the country and in this continent. There's going to be one team that's going to play solely as a team. No man is more important than the team. No coach is more important than the team. Team, the team, the team. And if we think that way, all of us, everything that you do, take into consideration what effect does it have on my team. Because you can go into professional football, you go anywhere you want to play after you leave here. You will never play for a team again. You'll play for a contract, you'll play for this, you'll play for that, you'll play for everything except the team. Think what a great thing it is to be a part of something that is the team. Go win it. Go win the championship again, but we're going to play as a team.
So there's a number of things that I really like about Bo's speech. The notion that no individual is more important than the team is just incredibly important in team science. And in fact, I say that one of the biggest dangers or toxins to team science are people that are narcissistic and that want more out of the team than they're willing to give to the team. And so that's really important. I think it is the case that a lot of people have actually never had the opportunity to work on really high functioning teams and don't even know what it's like to be on a high functioning team. But if you do know what it's like to be on a high functioning team, you don't ever want it to end. It's really sort of a very cool atmosphere. Certainly I myself, as the leader of the Chronic Pain and Fatigue Research Center, I think I spend a lot of my time enforcing the culture of the team and making sure that everyone is treating each other with respect, that no individual on the team, whether it's a staff member or a faculty member, is more important than anyone else on the team. And if you, as the team leader, provide that sort of space, it's much more likely the team is going to prosper. So the way we're constructed, we are in the Department of Anesthesiology, the Chronic Pain and Fatigue Research Center. In order to get a center approved at the University of Michigan or elsewhere, you have to show that you're doing work that's interdisciplinary and usually crosses departments, crosses schools or colleges, and we certainly do this. This is just the order in which people sort of joined our group. Dave Williams actually joined our group while we were still at Georgetown. The rest of the people that have joined our group, many of these individuals used to be mentees of ours before they became faculty. And one of the things I'm most proud about our group is that now, notwithstanding some of the people down at the bottom are still junior faculty, or Lars Fritz, who just joined the group, he's more senior, that about 7 or 8 of these individuals are now thought to be successful and independent, even though they are within the Chronic Pain and Fatigue Research Center. When I talk about building a team, one of the things I first say to people is, if you want to build a team where other faculty can become independent and successful, the first thing you have to do is not name the team after yourself. I cringe sometimes, and this is nothing negative about groups that say we work in the X lab, or i.e. the last name of the leader of the team. But if the Chronic Pain and Fatigue Research Center were the Clauw lab, I think it would have been a lot more difficult for all these individuals to become independent and autonomous if it seems as though you're working in someone else's research group. We work in the Department of Anesthesiology, where there are a number of other well-known pain researchers. Most of us in the Chronic Pain and Fatigue Research Center do more mechanistic research in chronic pain. A lot of our colleagues in the department do more work in the opioid or perioperative pain space, but we work closely with all of these individuals in our department. We also work very closely with a lot of individuals elsewhere at the University of Michigan, where we provide the core expertise in functional neuroimaging or quantitative sensory testing or neuroimmunology, and we're working with people in engineering and OBGYN and urology, but basically because pain happens to involve a lot of different clinical conditions. There's a lot of individuals interested in studying pain. We do collaborate widely with a lot of people that are seeing pain in different areas of the body, as well as with data scientists and other individuals here at the University of Michigan. And then finally, we do a lot of training. I think that one of the things that has made our group most successful as a research team has been that we have been extremely focused on the junior faculty on mentoring, identifying and mentoring really talented junior faculty. They bring all the cool new ideas into the group or into the team. And I think that I joke about this, that it's been sort of an accidental business model, that early on we embraced the notion that mentoring was important, training was important. And again, nearly all of the individuals at our faculty in our group started out as mentees in our group. We do mentor a lot of other people elsewhere at the University of Michigan. I've been the primary mentor for people on career development awards at other institutions. And one of the coolest grants that we have received is a grant we received about 3 or so years ago. It's called the HEAL Pain K-12, where we provide the mentoring and training for a lot of individuals doing pain research across the country. There's now 13 people appointed to this K-12. They stay at their home institution and have like a regular career development award, but they get their mentoring in pain from us at the University of Michigan. And then there's hybrid models where some of the mentoring team is at Michigan, some of the team is elsewhere. We collaborate a lot in NIH networks. I'm going to show you a couple NIH networks that we've been involved in because I think it's a really cool place to put different types of technology and gadgets. Certainly it's where we've identified both some cool gadgets as well as some cool collaborators that have spent a lot of time developing their gadgets that we can then later adopt. And then outside collaborators. So again, to finish the part about team science, some random advice for junior faculty. I don't think we talk to people enough when they're in training about where they want to be in team science. Do you want to be the leader of the team? Do you want to be the member of a team? That you're still the PI of grants, you're still independent and successful, but you really don't want to do what Dan Clauw does to lead a team. You don't want to have to fight for space. You don't have to want to fight for resources. It turns out the Department of Anesthesiology is incredibly generous to us. We don't really have to fight for resources, but nonetheless, there's a lot of individuals who, a lot of the faculty in our group, many of whom are sort of conflict averse and would rather just be in a group rather than be leading a group. You can also be a researcher that's not really interested in writing and getting grants, but you play an incredibly important role in team science. You know how to analyze data from actigraphy. You know how to analyze a certain amount of data. And you don't really want to be on one of the research tracks that require that you're the PI of grants, but you want to be on a track like the research investigator, the research scientist track, where people don't have to be the principal investigator of grants, but they're a very valuable member of team science. And then finally, you can be someone who's trained as a clinician who contributes to team science, but isn't writing or getting grants. I already talked about this, but narcissists are toxic to teams. You have to make sure they don't get in your team or they will. They get a single person that's narcissistic can destroy the culture of an entire team. It's extremely important when people get into teams that the junior people sort of pair up together. Most clinical researchers that are trained as clinicians need 2, 3 sort of PhD trained methodology people, and they can help each other a great deal in their careers. The PhD trained methodologists need clinicians that they can count on to recruit patients, to come up with good ideas, and the clinicians need the PhD trained methodologists just as badly. And when you're early in your training, I think you can be a little bit less intentional about precisely what topic you study, because I think being in successful teams is just a terrific training environment. Even if you don't, for example, end up going into pain, just learning how to do team science in a group like ours or another well-established group that's studying a certain set of conditions can be extremely helpful. One of the other things that I really strongly encourage people to do is to look for mentors outside the institution. I think a lot of people are a little timid about approaching people outside. It's like, oh, I'm not going to email Dan Clauw because he wouldn't never mentor me. He's too busy. It's like many of us that we enjoy mentoring so much that we rarely tell people that we can't mentor them, and we make the time for mentoring because because it's really fun and it's really the main thing that we do as we get on in our careers. Think of your research like a stock portfolio. You're going to do safer foundational research early and riskier research later. Don't try to take giant steps. Be entrepreneurial and opportunistic. I think, again, that's another thing that's helped us a great deal is if the NIH was interested in funding research on interstitial cystitis or urinary chronic pelvic pain, and they put out a big RFA, then we're going to compete for it. It wasn't something that we were inherently that interested in, but if that was where the money was, and if that's where the first big NIH network was going to be, then we could be a little bit flexible with respect to what pain condition we wanted to study. And then finally, it is just incredibly important to be perseverant and to work hard. We get defeated a lot in research, and you have to be willing and able to dust yourself up and get up and start over again. For the mentors, I think there's an epidemic of imposter syndrome. Nearly all of my mentees have been some type of imposter where they are better than they think they are. And what we're having to do is sort of prop them up and tell them that they're really good and help. And the other thing that teams do that people don't appreciate, I think everyone, when they talk about the value of working in a team, they tend to focus on the value of the higher quality science that would come out of a team. But I think one of the real benefits of being in a team is the buffering that occurs when you get the rejections and the failures that we often do in science, those grants and those manuscripts that are rejected. As soon as someone gets a grant or a manuscript that's rejected, the entire team rallies around them, tells them they're going to be fine, helps them write the next version of the grant. But there really is this strong culture of the team to help against the rejections that we get often in science. Those of us that are more senior often embrace and model failure because people, again, have the tendency to think that we've never had any failures or that we never had any grants that weren't funded. I often will pull out a list of the several $100 million in grants that I've written that didn't get funded because all people hear about when they do the introduction of you is all your grants that did get funded. And then an important thing that mentors do is really help mentees avoid rabbit holes. And technology and gadgets can be rabbit holes. I'll talk about this, but this is, I think, one of the most important responsibilities of a mentor is to make sure that whatever someone is working on, especially if they're early in their career or junior, is really going to be fruitful with respect to leading to the next grant and leading to the next training opportunity. And again, this seems little bit antithetic, but I think that it's as important when people are working in teams that you recruit for personality rather than for CV. And so now a little bit about pain. There's three mechanisms of pain. The mechanism on the left, nociceptive pain, is the one you're probably all most familiar with. You bang your thumb with a hammer, you have a surgical procedure, but there's some damage or inflammation out in the periphery that activates a nerve, and you're having pain because you're supposed to have pain because you have injured yourself and you're supposed to stop moving that area of the body. The second kind of pain is nerve pain or neuropathic pain, where you have a pinched nerve or a damaged nerve. But this third mechanism of pain wasn't really understood until about 30 or so years ago. This used to be called central sensitization. Centralized pain is now called nociplastic pain, but it's where the pain is really coming more so from the central nervous system rather than it being driven by some damage or inflammation out in the periphery. And we now know that this kind of pain is much more widespread. It's accompanied by a lot of other symptoms like fatigue and sleep, memory, and mood problems. And people will often not just be sensitive to pain, but they'll be sensitive to other types of sensory stimuli like the brightness of lights, the loudness of noises or odors. So this type of pain, this third type of pain, is what our group is best known for. We have done the work in a lot of these conditions that has shown that this central sensitization or this CNS-driven pain is the predominant type of pain in these conditions. And what's really most important clinically about identifying this is that this kind of pain that is coming from the brain obviously is not going to respond to things like surgical procedures or injections, and even classes of drugs like opioids don't work in this kind of pain. So it's been important to help clinicians identify when someone has this pain that's coming from the central nervous system. It's not just a pedantic interest, it's going to dramatically affect what's going to work and what's not going to work with respect to treatments. And so all of these conditions now are really thought to be this kind of pain, tension and migraine headache, fibromyalgia, irritable bowel, temporomandibular disorder, what used to be called interstitial cystitis is now called bladder pain syndrome, and even conditions like low back pain or endometriosis. You may think because of the title that most low back pain comes from a problem in the back and most endometriosis comes from implanted endometrial tissue, but it turns out that the majority of individuals with even these conditions like low back pain and endometriosis, their pain is coming more so from the central nervous system. And some of these individuals, the pain isn't quite as prominent. The fatigue, the sleep, and the memory problems are more prominent. These would be things like chronic fatigue syndrome, long COVID, would be examples of these kinds of syndromes. But all of these conditions are really closely related to each other. In fact, one of the terms is chronic overlapping pain conditions, because if someone has one of these conditions, they're more likely to have several of these conditions. The fundamental problem is the central nervous system is on fire, and these individuals are going to have pain all over their body because of a CNS problem. And these are some other possible chronic overlapping pain conditions that are related, including post-deployment syndromes, post-infectious syndrome that our group has studied a lot over the years. Again, this new term is used more and more, nosoplastic pain. We realize that these individuals have increased central nervous system pain processing as well as sensory processing. And we also know that they have some of these other CNS symptoms like fatigue, sleep, memory, and mood problems. Because of this, we measure a lot of these things. And so one of the gadgets that we've used a lot in our research is our different types of actigraphs, because measuring sleep more precisely using actigraphy, measuring activity more precisely using actigraphy can give you additional information to what you can get with patient-reported outcomes. But we write a lot about nosoplastic pain in different articles. This one's very densely cited if you really care. It's a Nature Reviews Neurology article. And then there's sort of two types of this kind of pain. One is that people that mainly have this kind of pain, and then the other are the people with conditions that start out as nociceptive pain, like autoimmune diseases like rheumatoid arthritis or lupus or sickle cell disease, that these people start by having pain that is due to active inflammation or active damage, but over time they had developed superimposed fibromyalgia pain or nociplastic pain that again needs to be treated in a different way. And so one of the analogies I started using a long time ago is that if the amount of pain that someone is experiencing is analogous to the loudness of an electric guitar, if someone has a problem in their knee, that's more of a guitar problem, that the sensory nerves here are being activated, and someone is, this for example, is someone with bone on bone, should have knee pain because there's no cartilage left in their knee anymore. But it turns out that this information from the periphery goes up through the spinal cord and the brain before it can be felt as pain. There's a lot of gating of this information. And so it turns out that one of the major determinants of whether you're going to have pain in a region of your body or not is not whether you have damage in that region of your body, it's what your amplifier is set at. Do you have a central nervous system that lets all this sensory information just go up to the brain to be felt as pain? Are you a pain sensitive person that has a high amplifier setting? Or are you a pain insensitive person that even though you have a knee x-ray that looks like this, you don't have any pain because this information isn't allowed to go up to the brain to be felt as pain? So this is the kind of work that we do using things like quantitative sensory testing and functional neuroimaging to try to infer like where the pain is coming from. So one of the first pain network that the NIH funded was called the MAP network, the multidisciplinary approach to chronic pelvic pain. This, like a lot of NIH networks, is a nationwide network. We had one of the 6 discovery sites at the University of Michigan. Even though I'm a rheumatologist, I was asked to chair this network because no urologist knew anything about pain. So for the first 5 years of the network, I was given the opportunity to chair this network, which was really a lot of fun. And the whole point of this network was to perform phenotyping in these individuals with chronic pelvic pain, which we called urinary chronic pelvic pain syndrome, and then look and see what different phenotypes match up with what different treatments, because we knew this was not all one type of pain and it wasn't going to respond to all one type of treatment. And so these individuals in these network studies often come in multiple points in time, these are often sort of longitudinal observational studies, and you do a bunch of stuff to them. You do a bunch of neurobiological studies, all sorts of different, and this is where a lot of gadgets come into play in these large NIH network studies, is different sorts of sensors or gadgets or things that can really draw inferences about the underlying mechanism of pain or how much someone's moving or what they're moving. In the map, just to sort of summarize what we found in the map studies, the first thing we found using very simple body maps is that a lot of people that came in with interstitial cystitis didn't only have pain in their pelvis, they had pain in their abdomen, or they had a more widespread pain phenotype like we see in individuals with fibromyalgia. But 60 or 70% of interstitial cystitis patients look a lot more like fibromyalgia patients if you really give them a body map and you ask them to check all the places that they have pain. And then sure enough, as we went on in the network and did functional brain imaging, the people with pelvic pain that was in their bladder alone looked a lot like the healthy controls, whereas the people with pelvic pain that had the more widespread pain phenotype on functional brain imaging looked almost identical to fibromyalgia patients. We also found that these individuals with interstitial cystitis were very sensitive to lights and noises and odors, and decided to double down on those findings. And then at the end of the network, we actually showed that the individuals with chronic pelvic pain that had pain confined to the bladder responded really well to treatments like physical therapy to the pelvic floor or things that were instilled into the bladder. Whereas those individuals that had the widespread pain phenotype, even though they were called interstitial cystitis, responded a lot better to classes of drugs that work in conditions like fibromyalgia, tricyclic drugs, gabapentinoids, serotonin norepinephrine reuptake inhibitors like duloxetine. This is the next NIH network that I had the opportunity to co-chair. This network is just ending now. It's called Backpack. And this network was a low back pain study that was not really at all interested in looking at whether new treatments worked in low back pain, but instead it was interested in looking at what phenotypic predictors predicted what was going to work, is i.e., for these treatments that we know work in a third of people with low back pain, like physical therapy, like duloxetine, like what can we do at baseline that's going to identify the phenotypic predictors of who's going to respond best to physical therapy, who's going to respond best to acupuncture, who's going to respond best to duloxetine, because we may be able to take treatments that right now work only in a third of people and get them to work in 2/3 of individuals just by being smarter about who we give these treatments to. And so in the first phase of the backpack studies, we gave individuals 4 different types of commonly used therapy for chronic low back pain. This is a type of cognitive behavioral therapy. This is a website. This is physical therapy. This is duloxetine. And the point of these studies was, again, not to pit one of these treatments against the other. It was to see what phenotypic predictors at baseline were going to say that someone is going to respond best to this type of CBT, to physical therapy, to duloxetine. And we used a smart design where we randomized people 2 different times. If they didn't respond well enough to the first treatment, they got responded to a second treatment. And so in these trials that we did at the University of Michigan in a big 14-site, multi-site trial, both of which have ended in the last 6 months or so, we were able to identify, just using simple patient-reported outcomes, subgroups of people that were a lot more likely and a lot less likely to respond to cognitive behavioral therapy, to physical therapy, to duloxetine, to websites. And a new trial that we just got, it was just announced in the University of Record, a $16.5 million grant, is essentially going to redo this big clinical trial, but what we'll be doing is randomizing people based on phenotype, and then randomizing based on random. And what we hope to show in this new study is that phenotype matters and that we can identify, using these phenotypic characteristics, the individuals that are going to respond well or less well to commonly used treatments. So here's like all the different things that we put in these backpack studies. We have 3 different types of biomechanical sensors in all of these individuals that are going through all of these treatments. They have all this biomechanical assessment. They get spine imaging, they get functional brain imaging, they get quantitative sensory testing. But you can see we're doing a whole bunch of different things to individuals at baseline to look at their phenotype, and this hopefully will predict a differential response. One of the gadgets that we use is from Bill Morris at Ohio State. This is a motion sensor that's a really cool motion sensor. Bill and his group have worked about 10 years developing and perfecting this motion sensor. It only takes about 6 minutes to put this on and to give someone do this rapid test. And we get a really good readout then from this test of like how well someone can move through different planes and different ranges of motion. You see here someone going through the motion sensor test right here, and then you get a readout here that basically tells you what's moving, what's not moving, and we're using this both as a phenotypic predictor. Can we, based on something that we identify at baseline, identify, for example, is someone more likely to respond to physical therapy if they have a certain biomechanical pattern here? But then we're also looking at this as an outcome measure. Is it if we give people certain treatments, can we see that their range of motion improves over time? Another thing that we do through our collaborative networks is we do a lot of different studies of people that are getting surgical procedures that were intended to relieve pain. We again do all these phenotypic measures, and we see which phenotypic measures can identify the individuals who are getting surgery for pain, because we think that the people who don't respond to these surgical procedures that are given for pain, like knee and hip replacement surgery or hysterectomy for chronic pelvic pain, we think these are the ones that have pain that are coming from the brain. And this is a test that Steve Hart in our group developed. It's a visual quantitative sensory test that actually, when people look at this, we vary the brightness of the light. And this test is to test how bright someone views a light to be, because in a lot of the other studies that we did in the map in our earlier studies, this hypersensitivity to light was a really strong signal that we were seeing that these individuals with nosoplastic pain are not just pain sensitive, they're more globally sensory sensitive to the bright lights, to loud noises, to odors. And this quantitative sensory test was incredibly powerful in some studies that we recently just completed. These are people that were getting knee replacement surgery. And if we just did that visual quantitative sensory test at baseline, the results of that visual QST alone predicted whether someone was going to get better after they got their knee or their hip replaced. The non-responders to surgery looked almost identical to fibromyalgia patients, and they viewed that light to be very bright, whereas the people who responded to surgery looked a lot more like the healthy controls. And so this visual QST measure is something that we've integrated into nearly all of our studies as sort of a gadget that we can use to identify people that have this kind of pain. And then finally, this is a really cool gadget that Steve Hart in our group and Grant Kruger in the College of Engineering developed. They developed this with SBIR funding. One of the advantages of the small business funding is they make you go through a lot of steps of talking to a lot of end users, making sure that there's billing codes that you can use to do this. But this is a really cool actigraph. We use actigraphs in a lot of our studies, but things like, and we use things like Fitbits or use the actigraphs that are in people's smartphones. But people don't wear their smartphones on their body all the time. Fitbits have to be taken off. This cool gadget that Grant and Steve developed is just almost like a little rubber band. When it's taken out of an envelope, the air makes it turn on automatically, and it has a battery that lasts for 30 days. So you don't have to pair it to a smartphone, which if you ever have tried to use an actigraph in research studies, one of the biggest challenges, especially if you have elderly individuals in your study, is your research assistants have to become very tech savvy and help pair things. This gadget doesn't have to be paired. It just collects data for an entire month, and then people put it back in an envelope and they mail it back and we get a readout of activity, really nice over an entire month. And so when these are produced at a bigger scale, we expect that this new actigraph will replace all the actigraphs that we've historically used just because of the usability of it and because people don't ever have to take it off like they do most of the other types of things. And then finally, this is in my last couple slides. This is another cool gadget that one of our mentees at Mount Sinai, Bridget Mueller, she's a headache and migraine specialist, and she's very interested in autonomic function. This is a gadget that not only measures heart rate variability, which is a good way of estimating parasympathetic tone, but it also has galvanic skin resistance, so you can get a measure of sympathetic tone as well. And so this is a cool ambulatory device to look at autonomic function. And Bridget's embedded this into a lot of her ongoing studies. And it's a pretty good way of measuring autonomic function in an ambulatory setting, but this, like any other gadget, it took her 2 to 3 years of starting to use this before she felt comfortable acquiring the data. And now the issue, and now that she has acquired the data, is really analyzing all the data. So I'll end with this slide that wearable and sensor data are being increasingly used in clinical and translational research. These are often really cool, bright, shiny objects. When I teach grant writing, I say that it's often nice to have a bright, shiny object on your Ames page, something cool or innovative. And often these gadgets or these sensors are an example. But there's a big but here. Each new device has a significant learning curve just to how to acquire or collect the data. There's a lot of like glitches with new gadgets and new devices. After you figure out all this, then you have to figure out how to analyze the data. And often it's a different group of individuals that are going to have to help you analyze your data than the people that made the gadget that's going to collect the data. And probably the biggest thing I would recommend, if you're a gadget developer, then I'm not really talking to you. I'm talking to someone that's a gadget user, an end user, is work with someone that's worked through these things rather than to try to do all these things yourself, because it seems like every step along the way here takes 2 to 3 times as long as you think it would take if you haven't ever done anything like this before, where you've developed a new gadget and tried to bring it all the way into using it in clinical research. So I'll stop there and take any questions people might have.